Hi everyone. Today we are going to talk about another topic of digestive system, which is stomach. So this is important one as it is being asked in exams as a question of 10 marks, 5 marks and 2 marks. If I'm talking about the question of 10 marks, so digestion in stomach, you have to describe the digestion in stomach in 10 marks. If I'm talking about the 5 marks question, then the functions of gastric juice, the composition of gastric juice, and pH value of the gastric juice. They are there are questions which are being asked generally. And if I'm talking about the 2 marks question, to secretion of the stomach, gastric lipase function, this kind of questions are being asked. So again, it is important. So let's start. First, we'll see the functional anatomy of the stomach. The stomach is a J-shaped enlargement of the GI tract directly inferior to the diaphragm in the abdomen. Stomach is a hollow organ situated just below the diaphragm on the left side in the abdominal cavity. Volume of empty stomach is 50 ml. Under normal conditions, it can expand to accommodate 1 liter to 1.5 liter of the solids and liquids. However, it is capable of expanding still further up to 4 liter. Parts of stomach. In humans, stomach has 4 parts: cardiac region, fundus, body or corpus, and pyloric region. So in this figure, we can see the different parts of stomach. This is the figure of stomach. This is the fundus part, upper part, then this is the body. This is the cardiac region, and this is the pylorus. Okay, cardiac region. Cardiac region is the upper part of the stomach where esophagus opens. The opening is guarded by a sphincter called the cardiac sphincter, which only opens towards stomach. This portion is also known as cardiac end. Fundus. Fundus is a small dome-shaped structure. It is elevated above the level of esophagus opening. Body or corpus. Body is the largest part of stomach, forming about 75 to 85% of the whole stomach. It extends from just below the fundus up to the pyloric region. Pyloric region. Pyloric region has 2 parts, antrum and pyloric canal. The body of stomach ends in antrum. Junction between body and antrum is marked by an angular notch called incisura angularis. Antrum is continued as the narrow canal, which is called pyloric canal or pyloric end. Pyloric canal opens into first part of small intestine, which is called the duodenum. The opening of pyloric canal is guarded by a sphincter called the pyloric sphincter. It opens towards duodenum. Stomach has 2 curvatures. One of the right side is lesser curvature and the other on the left side is a greater curvature. Structure of the stomach wall. Stomach wall is formed by 4 layers of the structures. Outer serous layer, which is formed by the peritoneum. Muscular layer made up of 3 layers of smooth muscle fibers, namely inner oblique, middle circular and outer longitudinal layers. Submucous layer formed by areolar tissue, blood vessels, lymph vessels and mesenteric nerve plexus. Inner mucous layer. Inner mucous layer lined by mucus secreting columnar epithelial cells. The gastric glands are situated in this layer. Under resting conditions, the mucosa of the stomach is thrown into many folds. These folds are called rouges. The rougi or rouges disappear when the stomach is distended after meals. Throughout the inner mucous layer, small depressions called gastric peats are present. Glands of the stomach open into these peats. Inner surface of mucus layer is covered by 2 mm thick mucus. Now we'll talk about the glands of stomach. Gastric glands. Glands of the stomach or gastric glands are tubular structures made up of different types of cells. These glands open into the stomach cavity via gastric peats. Classification of glands of the stomach. Gastric glands are classified into 3 types on the basis of their location in the stomach. First is the fundic glands or main gastric glands or oxyntic glands, which are situated in body and fundus of stomach. Pyloric glands present in the pyloric part of the stomach, and cardiac glands located in the cardiac region of the stomach. Now we'll see the structure of gastric glands. Fundic glands. Fundic glands are considered as the typical gastric glands. These glands are long and tubular. Each gland has 3 parts: body, neck, and isthmus. Cells of fundic glands. Chief cells or pepsinogen cells, parietal cells or oxyntic cells, mucous neck cells, enterochromaffin or EC cells, and enterochromaffin-like ECL cells. Parietal cells are different from other cells of the gland because of the presence of canaliculi. Parietal cells empty their secretion into the lumen of the gland through the canaliculi, but other cells empty the secretion directly into the lumen of the gland. Pyloric glands. Pyloric glands are short and tortuous in nature. These glands are formed by G cells, mucus cells, EC cells, and ECL cells. Cardiac glands. Cardiac glands are also short and tortuous in structure with many mucus cells. EC cells, ECL cells, and chief cells are also present in the cardiac glands. This is the structure of gastric glands. This is a gastric peat. Then you can see the mucous neck cell. In pink, there are chief cells, there are parietal cells, and the lumen of the gland. Enteroendocrine cells. Enteroendocrine cells are the hormone secreting cells present in the glands or mucosa of the gastrointestinal tract, particularly stomach and intestine. The enteroendocrine cells present in gastric glands are G cells, EC cells, and ECL cells. Functions of the gastric gland. Functions of the gastric gland is to secrete gastric juice. Secretory activities of different cells of gastric glands and enteroendocrine cells are listed. Okay, fine, we'll see the table first. Okay. If we are talking about the chief cells, so its secretory products would be pepsinogen, renin, lipase, urease, and gelatinase. If we are talking about the parietal cells, so secretory products of parietal cells would be hydrochloric acid, intrinsic factor of castle. Mucous neck cells and secretory product of mucous neck cells would be mucin. Then there is a G cells, and the secretory product is gastrin. Then there are EC cells which secretes serotonin, and there are ECL cells which secretes histamine. Now we'll see the functions of stomach. First function of stomach would be the mechanical function. It includes many functions in that, like storage function. Food is stored in the stomach for a long period, for 3 to 4 hours, and emptied into the intestine slowly. The maximum capacity of stomach is up to 1.5 liter. Slow emptying of stomach provides enough time for proper digestion and absorption of food substances in the small intestine. Formation of chyme. Peristaltic movements of stomach mix the bolus with gastric juice and convert it into the semi-solid material known as chyme. Then there is a digestive function, protective function, and hemopoietic function. We'll talk about these three functions when we'll talk about the functions of gastric juice. Then there is excretory functions. Many substances like toxins, alkaloids, and metals are excreted through gastric juice. Now we'll see the properties and composition of the gastric juice. Gastric juice is a mixture of secretion from different gastric glands. Properties of gastric juice. The volume of the gastric juice would be 1200 ml per day to 1500 ml per day. Gastric juice is highly acidic with a pH of 0.9 to 1.2. Acidity of gastric juice is due to the presence of hydrochloric acid, and the specific gravity would be 1.002 to 1.004. Composition of the gastric juice. Gastric juice contains 99.5% of water and 0.5% of solids. Solids are organic and inorganic substances. In this chart we can see gastric juice is made up of water, which is 99.5%, and solids are 0.5%. If we are talking about the solids, there would be organic substances and there would be inorganic substances. In organic substances there would be enzymes and there would be other organic substances. In enzymes there would be pepsin, rennin, gastric lipase, gelatinase, and urease. And in other organic substances there would be mucus and intrinsic factor. In inorganic substances there would be hydrochloric acid, sodium, calcium, potassium, bicarbonate, chloride, phosphate, and sulfate. Now we'll see the functions of the gastric juice. First function is the digestive function. Gastric juice acts mainly on proteins. Proteolytic enzymes of the gastric juice are pepsin and rennin. Gastric juice also contains some other enzymes like gastric lipase, gelatinase, urease, and gastric amylase. Pepsin. Pepsin is secreted as inactive pepsinogen. Pepsinogen is converted into pepsin by hydrochloric acid. Optimum pH for activation of pepsinogen is below 6. Action of the pepsin. Pepsin converts protein into proteose, peptones, and polypeptides. Pepsin also causes curdling and digestion of the milk. Gastric lipase. Gastric lipase is a weak lipolytic enzyme when compared to pancreatic lipase. It is active only when the pH is between 4 to 5 and becomes inactive at the pH value of below 2.5. Gastric lipase is a tributyrase and it hydrolyzes tributyrine into fatty acid and glycerols. Actions of other enzymes of gastric juice. Gelatinase. It degrades type 1 and type 5 gelatin and type 4 and type 5 collagen, which are proteoglycans in meat, into peptides. Urease. It acts on urea and produces ammonia. Gastric amylase. It degrades starch, rennin, curdless milk. Second function of the gastric juice would be hemopoietic function. Intrinsic factor of gastrin secreted by parietal cells of the gastric glands plays an important role in erythropoiesis. It is necessary for the absorption of vitamin B12 from GI tract into the blood. Vitamin B12 is an important maturation factor during erythropoiesis. Absence of intrinsic factor in gastric juice causes deficiency of vitamin B12, leading to pernicious anemia. Protective function. Function of mucus. Mucus is a mucoprotein secreted by mucous neck cells of the gastric glands and surface mucus cells in fundus, body, and other parts of the stomach. It protects the gastric wall by the following ways. Mucus. It protects the stomach wall from irritation or mechanical injury by virtue of its high viscosity. It prevents the digestive action of pepsin on the wall of the stomach, particularly gastric mucosa. It protects the gastric mucosa from hydrochloric acid of gastric juice because of its alkaline nature and its acid combining power. Functions of hydrochloric acid. Hydrochloric acid is present in the gastric juice. It activates the pepsinogen into pepsin. It kills some of the bacteria entering the stomach along with food substances. This action is called the bacteriolytic action. It provides acid medium which is necessary for the action of hormones. Secretion of the gastric juice. Secretion of the pepsinogen we'll talk about. Pepsinogen is synthesized from amino acid in the ribosomes attached to endoplasmic reticulum in chief cells. Pepsinogen molecules are packed into zymogen granules by Golgi apparatus or Golgi apparatus. When zymogen granule is secreted into stomach from chief cells, the granule is dissolved and pepsinogen is released into gastric juice. Pepsinogen is activated into pepsin by hydrochloric acid. Secretion of hydrochloric acid. According to Davenport theory, hydrochloric acid secretion is an active process that takes place in the canaliculi of the parietal cells in gastric glands. The energy for this process is derived from the oxidation of the glucose. Carbon dioxide is derived from metabolic activities of parietal cell. Some amount of carbon dioxide is obtained from blood also, and it combines with water to form the carbonic acid in the presence of carbonic anhydrase. This enzyme is present in high concentration in parietal cells. Carbonic acid is the most unstable compound and immediately splits into hydrogen ion and bicarbonate ion. The hydrogen ion is actively pumped into the canaliculus of the parietal cell. Simultaneously, the chloride ion is also pumped into the canaliculus actively. The chloride is derived from sodium chloride in the blood. Now the hydrogen ion combines with chloride ion to form the hydrochloric acid to compensate the loss of chloride ion. The bicarbonate ion from parietal cells enters the blood and combines with the sodium to form the sodium bicarbonate. So in this figure, we can see the secretion of hydrochloric acid in the parietal cells of the gastric gland. So this is a graphical illustration of the secretion, what we have talked about further. I think I should not have to go for it again. Okay, fine, let's try this. Okay, so as we have talked about, the carbon dioxide is derived from the metabolic activities of parietal cells. Some amount of carbon dioxide is obtained from the blood also. So it combines with the water to form the carbonic acid in the presence of the carbonic anhydrase. So here the carbon dioxide combines with the H2O in the presence of carbonic anhydrase and converts into the carbonic acid. This enzyme is present in high concentration in parietal cells. Carbonic acid is the most unstable compound and immediately splits into hydrogen ion and bicarbonate ion. So you can see this hydrogen ion and bicarbonate ion. The hydrogen ion is actively pumped into the canaliculus of the parietal cells. So here in this figure we can see the canaliculus. Simultaneously, the chloride ion is also pumped into the canaliculus actively. So chloride ion is also here. Here is a hydrogen, here is a chloride. Now the hydrogen ion combines with chloride ion to form the hydrochloric acid. As we can see here, hydrochloric acid. To compensate the loss of chloride ion, the bicarbonate ion from parietal cell enters the blood and combines with the sodium to form the sodium bicarbonate. So this phenomena is, we can see here. This is the ECF, and the entire process is the secretion of hydrochloric acid. Factors stimulating the secretion of hydrochloric acid: gastrin, histamine, and vagal stimulation. Factors inhibiting the secretion of the hydrochloric acid: secretin, gastric inhibitory polypeptide, and peptide YY. Regulation of the gastric secretion. Regulation of gastric secretion and intestinal secretion is studied by some experimental procedures like Pavlov pouch method, Hedinda pouch, Bickel pouch, Farrell and Ivy pouch, sham feeding, but we are not talking about that because it is not necessary for you. So we are directly going to the phases of the gastric secretion. Secretion of gastric juice is a continuous process, but the quantity varies depending upon time and stimulus accordingly. Gastric secretion occurs in the 3 different phases: cephalic phase, gastric phase, and intestinal phase. So this question may be asked in 5 marks in exam, so it is important. In human beings, a fourth phase called interdigestive phase exists. Each phase is regulated by neural mechanism or hormonal mechanism or both. First, we'll talk about the cephalic phase. Secretion of gastric juice by the stimuli arising from head region, cephalus, is called the cephalic phase. This phase of gastric secretion is regulated by the nervous mechanism. The gastric juice secreted during this phase is called the appetite juice. During this phase, gastric secretion occurs even without the presence of food in stomach. The quantity of this juice is less, but it is rich in enzymes and hydrochloric acid. Nervous mechanism regulates the cephalic phase through reflex action. And 2 types of reflexes occur: unconditioned reflex and conditioned reflex. Unconditioned reflex. Unconditioned reflex is an inborn reflex. When food is placed in mouth, salivary secretion is induced. Simultaneously, gastric secretion also occurs. Stages of reflex action. Presence of food in the mouth stimulates the taste buds and other receptors in the mouth. Sensory impulses or afferent impulses from mouth pass via afferent type of nerve fibers of glossopharyngeal and facial nerves to amygdala and appetite center present in the hypothalamus. From here, the efferent impulses pass through dorsal nucleus of the vagus and vagal efferent nerve fibers to the wall of the stomach. Vagal efferent nerve endings secrete acetylcholine, which stimulates the gastric secretion. Conditioned reflex. Conditioned reflex is a reflex acquired by the previous experience. Presence of food in the mouth is not necessary to elicit this reflex. The sight, smell, hearing of or thought of the food, which induce the salivary secretion, induce gastric secretion also. Stages of reflex action. Impulses from the spatial sensory organs pass through efferent fibers of the neural circuits to the cerebral cortex. Thinking of food stimulates the cerebral cortex directly. From cerebral cortex, the impulses pass through dorsal nucleus of the vagus and vagal efferents and reach the stomach wall. Vagal nerve endings secretes acetylcholine, which stimulates the gastric secretion. Experimental evidences to prove the cephalic phase. Unconditioned reflex of gastric secretion is proved by sham feeding along with Pavlov pouch. After vagotomy, sham feeding does not cause gastric secretion. It proves the importance of the vagus nerve in the phase. Okay, we are not talking about this. Gastric phase. Secretion of gastric juice when food enters the stomach is called the gastric phase. This phase is regulated by both nervous and hormonal control. Gastric juice secreted during this phase is rich in pepsinogen and hydrochloric acid. Mechanisms involving gastric phase are nervous mechanism through local mind trick reflex and vagal reflex. Hormonal mechanism through gastrin stimuli which initiate these two mechanisms are distension of the stomach and mechanical stimulation of gastric mucosa by bulk of food, and chemical stimulation of gastric mucosa by the food contents. Nervous mechanism. Local myenteric reflex. Local myenteric reflex is a reflex elicited by the stimulation of the myenteric nerve plexus in stomach wall. After entering stomach, the food particles stimulate the local nerve plexus present in the wall of the stomach. These nerve fibers release acetylcholine, which stimulates the gastric glands to secrete a large quantity of the gastric juice. Simultaneously, acetylcholine stimulates G cell to secrete gastrin. Vagovagal reflex. Vagovagal reflex is a reflex which involves both afferent and efferent vagal fibers. Entrance of bolus into the stomach stimulates the sensory nerve endings of vagus and generates sensory impulses. These sensory impulses are transmitted by sensory fibers of vagus to dorsal nucleus of the vagus, located in medulla of brainstem. This nucleus is in turn sends efferent impulses through the motor fibers of vagus back to stomach and cause secretion of the gastric juice. Since both afferent and efferent impulses pass through vagus, this reflex is called vagovagal reflex. Hormonal mechanism. Gastrin is a gastrointestinal hormone secreted by the G cells which are present in the pyloric glands of the stomach. Small amount of gastrin is also secreted in mucosa of upper small intestine. In fetus, it is also secreted by islets of Langerhans in pancreas. Gastrin is a polypeptide containing G14, G17 or G34 amino acids. Gastrin is released when food enters stomach. Mechanism involved in the release of gastrin may be the local nervous reflex or vagovagal reflex. Nerve endings release the neurotransmitter called gastrin releasing peptide, which stimulate the G cells to secrete gastrin. Actions of gastrin on gastric secretions. Gastrin stimulates the secretion of pepsinogen and hydrochloric acid by the gastric glands. We are not talking about the experimental evidences. Intestinal phase. Intestinal phase is the secretion of gastric juice when chyme enters the intestine. When chyme enters the intestine, initially the gastric secretion increases but later it stops. Intestinal phase of gastric secretion is regulated by the nervous and hormonal control. Initial stage of intestinal phase. Chyme that enters the intestine stimulates the duodenal mucosa to release gastrin, which is transported to stomach by blood. There it increases gastric secretion. Later stage of the intestinal phase. After the initial increase, there is a decrease or complete stoppage of the gastric secretion. Gastric secretion is inhibited by two factors: enterogastric reflex and GI hormones. Enterogastric reflex. Enterogastric reflex inhibits the gastric secretion and motility. It is due to the distension of the intestinal mucosa by chyme or chemical or osmotic irritation of intestinal mucosa by the chemical substances in the chyme. It is mediated by the myenteric nerve, plexus and vagus. Gastrointestinal hormones. Presence of chyme in the intestine stimulates the secretion of many GI hormones from intestinal mucosa and other structures. All these hormones inhibit the gastric reactions. Some of these hormones inhibit the gastric motility also. GI hormones which inhibit gastric secretion: secretin, secreted by the presence of acid chyme in the intestine; cholecystokinin, secreted by the presence of chyme containing fats and amino acid in the intestine; gastric inhibitory peptide, which is called GIP, which is secreted by the presence of chyme containing glucose and fats in the intestine; vasoactive intestinal polypeptide, which is called VIP, secreted by the presence of acidic chyme in intestine; and peptide YY, which is secreted by the presence of fatty chyme in intestine. In addition to these hormones, pancreas also secretes a hormone called somatostatin during intestinal phase. It also inhibits gastric secretion. This is how this is how enterogastric reflex and intestinal hormones collectively apply a strong brake on the secretion and motility of stomach during intestinal phase. So in this figure you can see the graphical illustration, or you can see the chart for 3 phases like cephalic phase, gastric phase and intestinal phase, what you have talked about. And this is the 4th stage, which is interdigestive phase. As we have talked earlier, that there is a 4th phase in humans. Secretion of small amount of gastric juice in between meals is called interdigestive phase. Gastric secretion during this phase is mainly due to the hormones like gastrin. This phase of gastric secretion is demonstrated by Farrell and Eve pouch. Factor influencing gastric secretion. Gastric secretion is also influenced by some factors which increase the gastric secretion by stimulating gastric mucosa, such as alcohol and caffeine. So this was all about the stomach. The phases of gastric secretion is one of the frequently asked question in exams, or description of digestion in stomach in 10 marks. So this was all about today's topic. If you are having any query, let me know.